Hello friends, welcome again on this unique platform of e-learning. Our second topic of discussion is polymerase chain reaction and its application. In present scenario, when corona is making havoc, you might to know that this polymerase reaction machine, or that is called PCR, is the only device available to detect it. So this is a very important machine for any era of molecular biology. When we start with this topic, you might want to start with certain historical point of view. 1869, the first DNA is isolated, that is nucleus. Almost 100 years later, for the first time, we are able to amplify it in vitro condition. In 1988, in vitro amplification occurred with the help of thermostable tagged DNA polymerase, and within 1 year later, this gene for tagged DNA polymerase is cloned in E. coli, and now we are having our tagged DNA polymerase, recombinant tagged DNA polymerase isolated from E. coli. So when you talk about PCR, it is not a discovery. Actually, it is an invention where with the help of very special DNA polymerase enzyme, a short segment of DNA, about 100 to 10,000 base pairs, can be amplified with the help of primer. And for this invention, Carey Mills got Nobel Prize in 1993 in chemistry. This PCR has got very fame and name. Science magazine in 1989 described DNA polymerase as a molecule of the year. So such a huge impact has been made by this molecule on molecular biology. When a development point of view, we think initially the PCR was not like as today's PCR. Initially, they have three water baths, having the first water was having temperature of 94, the second water was with temperature of around 50, and third water bath with temperature of 72 degrees Celsius. And they use cleanout fragment for polymerization process. What they did, they make a reaction mixture, put the tube in the first water bath with 94 degrees Celsius for a particular period of time, and they remove the tube and kept in another water bath for annealing of primer. And once the annealing occurs, they again transfer the tube to third water bath and they add cleanout fragment. So the extension of primer takes place. And this one cycle once completed, they again go back for 94 degrees Celsius or first water bath where denaturation is occur. While doing so, the cleanout fragment added at the third stage is going to degrade because it is a heat liable. So every time they have to add a DNA polymerase or cleanout fragment, so it is a tedious job, and many times while doing so, the nucleus might enter and the whole reaction will go in vain. So it is not a great idea. Later on, in 1988, from the Yellowstone National Park, they isolate a bacterium from the hot spring whose temperature is more than 100 degrees Celsius, and that bacterium is able to survive. It means he has a DNA polymerase which is capable of working beyond 100 degrees Celsius, and that polymerase nowadays called as a tagged DNA polymerase, and as it is isolated from Thermus aquaticus. Nowadays, we will have a programmable thermal cycle which using a thermostable tagged DNA polymerase. So basically, PCR is aimed to amplify a specific region of DNA or to make multiple copies of gene so that they can be from a very minute source of DNA. So this is a particular setup of PCR. This is a PCR machine where you are going to keep your sample, and once the PCR is over, you fractionate this sample on agar gel electrophoresis method, and that utilized with the help of transcellumeter or gel document existing, and this result they are going to store in the form of final product. So what reagent you need to go for PCR? The first one is buffer. Buffer is required for optimum activity of enzyme, and basically it contains TCl and KCl. Then you need to have magnesium chloride, which ranges from 1 millimolar to 4 millimolar. Generally, 1.5 millimolar is the optimum concentration used in maximum PCR reaction. Then DNTPs, 4 DNTPs, A, T, G, C, they are added in a concentration of 200 millimolar. Then primers, you need to add primers around 100 nanometer, 100 nanometer to 1 micromolar concentration, and DNA polymerase, that is tag DNA polymerase, which is thermostable, generally which is used in a concentration of 1 unit to 4 units, but optimum concentration of 1 unit is okay, and you need to add DNA as a template in the concentration of 10 picogram to 101 microgram, and generally 20 nanogram is okay. So these all components you need to mix in a tube, and that tube is to be placed in PCR machine to complete the polymerase chain reaction. Basically, this whole process is divided into 3 steps. The first step is, once you added DNA, you need to separate it. Basically, the 2 terms are used, that is renaturation and denaturation. You need to know about renaturation and denaturation. The separation of fragments or separation of DNA is called denaturation, and joining of fragment is called renaturation. So if you expose the DNA to 94 degrees Celsius, the hydrogen bond present between the bases, they are separated, and you will get 2 separate strands. Then you down the temperature around 50 degrees Celsius, which is the optimum temperature for primer annealing. At that temperature, primer find out their complementary strand of DNA, and they are going to bind at a very precise location. Once they bind at very precise location, the DNA polymerase bind at the primer 3' end, and that bring out the complete synthesis of new strand by adding complementary nucleotides. This is how exponential growth of one segment of DNA is shown here. Copies of gene when exposed to first cycle, 2 copies or 4 copies means 2 template DNA having 2 strands, they are going to form 4 strands. Ultimately, after 35 cycles, you will got exactly 68 million copies of the template DNA that you are using. So it is an exponential growth of a sequence is occurs. So what are the templates for PCR? When you go for PCR reaction, you need to add DNA, and that DNA can be isolated from these various templates. The first is dried blood. From any crime scene, you can isolate a stain of blood, and from that blood, you are able to isolate DNA. Later on, semen sample, vaginal swab, single hair with root follicle, fingernail scraping, having certain skin tissues, then insects in umber, Egyptian mummies, they can be, from that DNA can be isolated. Buccal swab and toothbrush are some of the sources from where you can easily isolate DNA, and that isolated DNA acts as a template for PCR. Many times, friends, you correlate with the PCR with your replication. In replication, you know that one DNA is converted into two strands of DNA. Same thing occurs here. In replication, origin of replication is there, I is formed, then primer bind, and that is going to extend further. This is a replication course where helicase is there, which continuously separate DNA. I had the replication 4 with the help of topoisomerase, then single-stranded binding protein bind that kept these strands intact. Then polymerase is going to bind. Then primase is bind on the lagging strand. That primase is synthesized Okazaki fragments, and ultimately 2 strands are synthesized, lagging strand and leading strand. This is very similar to your polymerase, but how are the functions of replication achieved with PCR? We have to correlate by comparing the things. So the first function is melting of DNA. In PCR, the melting of DNA is achieved with the help of heat, while in case of replication, that is achieved with the help of enzymes like helicase, single-stranded binding protein, and topoisomerase. Then polymerization of DNA. Polymerizing DNA is occurred in PCR with the help of tag DNA polymerase, while in enzymes like DNA polymerase 1, 2, 3 in bacteria, and alpha, beta, delta, gamma, epsilon in eukaryotes. Then providing primer. You have to add artificially primer in PCR mixture, while in DNA replication, primase enzyme provide the primer. Joining of nicks is not available or not needed in PCR, where it is possible with the help of ligand. So here you can see how nucleic acids behave at different temperature. Initially, once the temperature rises at 94 degrees Celsius, the template is separate. So denaturation occurs and the two strands of DNA get separate. Then you lower down the temperature, that is annealing temperature, where the primer which are present in mixture, they bind to specific location on DNA. So this is the binding of primer, and once the primer binds to complementary position on template, you again rise the temperature around 72 degrees Celsius, where extension is brought. So denaturation, annealing, and extension, these three steps bring about the one cycle of PCR. And once that occurs, this is the completion of one cycle. Again, you go for melting with the help of rising the temperature to 94 degrees Celsius. Second cycle begins. Again, with the help of heat, two strands get separated. Again, the cycle gets repeated. Here again primer bind that bring about the extension, and ultimately two strand converted into four strand. Now here you just neglected the above and bottom strand that include the parental strand and focus on middle two strand with very short segment of DNA. When you go for third cycle, you will again go for first step, that is melting of DNA, then annealing of primer. Here annealing of primer occurs, and extension at 72 degrees Celsius. Ultimately, you will find the fragments of definite length are formed. And this is how a multiple fragment of definite length are formed when you increase the number of cycles. Here you will find the cycle numbers are given 0, 10, 15, 20, 25 and 35, and when these are the amplicons form after the cycle, you will find the band width is very high in 30 cycle PCR as compared to 0, 10 and 25. It means the some number of cycle increases, the bandwidth is going to increase or amplicon size is going to increase. This is all about the basics of principle. But friends, it's not that much easy to perform the PCR. Every time when you subject for any material or amplification, you need to optimize the condition. And the first optimization is buffer. Basically, the buffer contains KCl and trees. But now in the market, the pre-mixed buffer is available. So basically, nowadays, no one is going to make their own buffer. Then optimization second factor is MgCl2. MgCl2 is required for primer binding, and if you add excess magnesium chloride, that leads to non-specific binding, and if the concentration is low, the yield is reduced. The third one is primer designing. You need to have a primer which is very specific for complementary sequence. Generally, for REPD, they use 10 base pair of primer that is called decamer. And if you have a gene, they use very specific primer that is called as an oligomer, which ranges from 18 to 30 base pair nucleotide. And the temperature of primer binding is varies from 50 to 70. You need to justify or make sure what exact temperature will be there to specifically binding the primer. Then the third condition is you need to have GC content in the range of 40 to 60. And when you deal with eukaryote, the GC content should be more than 60%. And the third end of the primer should be always end with G and C. And it should be taken care that the internal sequence will not be complementary to each other. Otherwise, primer will refold and rejoin with itself. So these are the certain conditions you have to follow while designing the primer. Nowadays, there are certain softwares are there. With the help of that, you can design your own primer for particular gene. Then fourth one is the cycling condition. You have to determine the denaturation temperature that certain DNA polymerases require initial denaturation, that is called as a hot start. Then annealing temperature is important, that can be calculated with 4 into 4 G plus T plus 2 into A plus T, or by using primer software you can determine the annealing temperature. And decreasing annealing temperature result in non-specific binding, while increasing in the annealing temperature result in the reducing yield. This is the fourth condition for PCR optimization. And fifth one is cycle number. You have to determine how many cycles you want to go for PCR. Generally, they ranges from 25 to 40. It depends upon the half-life of the tagged DNA polymerase. So it is generally 30 minutes for tagged DNA polymerase at 95 degrees Celsius. It means that if you go for beyond 30 cycles, tagged DNA polymerase will not give you efficient results. So in summary, you need to have primer length, so that should not exceed 30. Then the TM, that is annealing temperature, is around 60 degrees Celsius. Primer should have GC content ranging from 40 to 60, and primer should always treat that with G or C to increase the efficiency of primer. So these are certain the applications of PCR. The first application is cellular cloning. Initially, cloning is used to amplify a segment of gene with the help of bacterium. The plasmid is taken, the plasmid is cut with the help of nucleoside. The foreign DNA also cut with recombinant DNA molecule. They merge and they transfer into the host cell, and they allow to amplify and magnify, reproduce, and later on the number of cell increases the number of genes. So here that is not needed. You directly amplify or with the help of a cellular condition. Second is, it is used as a diagnostic tool. In many disorders, where genetic disorders are there or infections are there, PCR used as a diagnostic tool to detect the infection. Then in genetic disorders, the change in genetic structure, it might be a single base that can be detected with the help of PCR. That is also used in genetic fingerprinting. The product of PCR is acts as a fingerprint for certain species or varieties, and it is highly used in forensic science, where a very small fragment of DNA is available at crime scene. That fragment of DNA isolated and amplified with the help of PCR, and ultimately is used to solve the crime scene. So this PCR is a very vital tool in nowadays biomolecular biology laboratory and occupying in very facet of life. Thank you very much.